Good afternoon, everyone. Thank you for coming to Joe thesis defense and thesis seminar. So it's quite easy to write for Joe's introduction. So today I'm just going to mention 3 firsts that's about Joe in my lab. Joe is the first PhD student coming to my lab, not for my own department, the Department of Biochemistry and Molecular Biology, but he is fully integrated to our department at multiple levels. He was part of the NCI training grant. He will serve as a teaching assistant for 3 years in our introductory course of molecular biology. And also he is one of the first inaugural awardee of Brian D. Crawford scholarship that is in my department. Joe is also the first student in my lab that's got an external fellowship. And we were very disappointed when we applied it, and then we got a 6th percentile grant. So for those who do not know what NIH grant means, when it's 6th percentile, say there are 100 applicants, you are the top 6, but you still did not get any money. So we, of course, we were very disappointed about that. But I think due to our persistence, and then also Joe's have a lot of updates that can send to the NIH. And finally, we got one of the very last NIH F31 predoctoral fellowship from the NIGMS. So the last first I want to say is that he is the first PhD student. I think that, I hope at least, that one day this discovery that we are going to, he is going to talk to you about later today would be in the textbook. I think that would be quite exciting. So if I really want to list a lot of different firsts, that could be quite a long list. Some are quite expected because who Joe is. Some are totally unexpected. But I will save it another time when we reminisce together. But then I just want to, instead, I want to sum up with 3 S. That's how I remember Joe. The first one is that Joe is super efficient. I'm not talking about normal efficiency. So when we discovered this pathway in 2016, I think still now we're still counting some construct we made recently only. I think we made about 200 different DNA constructs and another 200 different lockout or lockdown cell lines and over tens of thousands of PCR reactions. So you can talk about a lot of organizations still there. The next S is service. Joe is a great lab citizen, not just great, exemplary citizen. He makes sure the lab compile all the safety procedure. He's the person who do the lab order. And then he is also the person that makes sure that help organize when there is some equipment breakdown, he'll go help either find a vendor to repair it or he himself fix it. So I take this opportunity to say thank you. The last S is SRD. This stands for the pathway that we discovered. So that's structured RNA-mediated decay. So without further ado, please join me to wish Joe all the best in FDA. And also let's get to today's seminar get started, which is entitled Highly Structured 3' Untranslated Region Mediate RNA decay. Joe?
Thank you very much for that kind introduction, and thanks to all of you for coming out today. It's great to see so many people here. So like Andy said, I'm going to be talking about this project and this discovery I've been working on in his lab for the last 4 years. I just want to start with the central dogma and how, when you think about biology, it's the DNA, makes the RNA, makes the protein. And it's very important that this process is tightly controlled and everything is in perfect balance. Otherwise, you get disease, cancer, certain problems. So it's very important to tightly regulate all of this. And today what I'm going to be talking about is the RNA in terms of you need to be able to properly regulate that. And one way to regulate that is through RNA decay. Typically, when you think about mRNA decay, I just have some examples here. Just to orient everyone, we have the 5' cap, the 5' UTR. We have the coding region that translates into the protein, as well as the 3' UTR. And the 3' UTR is the main point of interest, as this is one of the main sites for regulating the decay of the RNA. And usually when you think about RNA decay, most decay pathways focus on sequence-specific features. So for example, in the top here, there are proteins that can recognize these AU-rich elements, the AREs, and they can recognize these ANU nucleotides, bind to them, and destabilize this transcript for decay. The other one that's very well characterized are the microRNAs, and these are these small RNAs that are also able to recognize the nucleotides within the 3' UTR to regulate the decay as well. However, besides just looking at the sequence, there are other mechanisms to degrade the RNA, and there are some things, such as the iron-responsive elements, in which within the 3' UTR, very well-defined structures can form within this through RNA-based pairing. And in this example, what happens is when these RNA structures are available to the cell, it is rapidly degraded. However, if these proteins, these IRPs, are able to bind to these very specific hairpins, they can initiate RNA decay, or protect the RNA from decay. But today, what I'm going to be talking about is a new form of RNA regulation. And these are the 3 main points I'll be talking about. I'm going to first talk about the proteins involved in the decay pathway, kind of go into more details on how it's decaying the RNA. And lastly, the thing that's most different about it is rather than being based on sequence-specific features or well-defined structures, it is actually based on overall structure, and I'll go into that more later as we get into it, what that really means. So first, getting into the proteins that are involved in this decay pathway. Where my luck really started was looking into this protein UPF1. This protein has been heavily characterized in various RNA decay pathways, most specifically in terms of nonsense mediated decay. But what people have found previously, when they looked at where UPF1 binds on mRNAs, is when you look at the top here, when you go past the stop codon into the 3' UTR, you can see there's an increase in the signal here suggesting that UPF1 is binding specifically to the 3' UTR. But the only reason it's binding there is due to translation, because when they inhibit translation with cyclohexamide, you now find UPF1 binding throughout the transcript. So people now really believe that UPF1 can bind non-specifically to RNAs. And then what this means in terms of decay is people have shown that when you have very long 3' UTRs, UPF1 is more susceptible to, or the mRNA is more susceptible to degradation, where the short ones are less susceptible. However, when you look genome-wide, you see that this doesn't really hold up very well. There's many short 3' UTRs that are regulated and many long 3' UTRs that are not. So where we thought might be a better way to explain it is that RNA structure. And one reason for that is there are some studies that do some single molecule imaging in which they were able to show that UPF1 is a double-stranded RNA binding protein and is a highly processive helicase. So what that means is UPF1 can bind to the protein here, and then it can start unwinding that RNA structure, and it will keep unwinding that RNA structure until it completes it, and once it's completed, it can release. So there's something about UPF1 being able to unwind these RNA structures that we thought could be important in terms of activating UPF1. So the first question I wanted to ask is, does UPF1 preferentially bind to these highly structured 3' UTRs? So to do that, I had some help from an undergraduate, very talented undergraduate, Claire, who was able to extract all the sequences for all 3' UTRs from the NCBI-RefSeq database. And what we can do with those then is use predictive programs to fold them and get an idea of how structured they really are. And I just want to take a minute, when you think about structuredness, the way that's easiest to think about it is the more double-stranded it is, the more base pairing you have going on, the more structured it is. But when you have more things like hairpin loops and single-stranded regions, the less structured it is. So then when you look at the distribution of all 3' UTRs, you can see on the left would be the ones that have very little structure and they're mostly single-stranded, whereas you get up to the 0.5, they're much more structured. So those are the ones that's going to have a lot more double-stranded RNA within the 3' UTR. So what we can do then is look, based on UPF1 binding, we can look from 4 different studies to see is there any preference to binding to these highly structured regions. And with this cumulative distribution frequency, you can compare distributions. And what you can probably notice is you're not really seeing anything going on, as it seems like wild-type UPF1 is able to bind non-specifically, as everyone has said before. But what we were able to do is the study by Lee et al. Had an additional factor, which helped us kind of better understand why it can bind non-specifically. And the reason it can bind non-specifically is if you look at a wild-type UPF1, is it can bind to both single-stranded and double-stranded regions. And the reason it can get into these more structured regions is because of that helicase activity. So when it has helicase activity, it can bind throughout the 3' UTR. However, if you mutate the helicase activity so it can no longer unwind it, what you might see then is that UPF1 can still bind to these single-stranded regions, but it's unable to get into these more structured regions. So if we then reanalyze this data where we look for regions in which wild-type UPF1 can bind, and the helicase mutant can't bind, maybe these will show a difference. So when you look at this, or you're looking at the helicase dependent, so that's 3' UTRs that have these wild-type UPF1 enriched regions, what you can now see is you do see this shift to the right. And what this shift to the right is showing is that the helicase-dependent UPF1 bound transcripts are much more structured on average. So with the ability to unwind these RNAs, they can more preferentially associate with those highly structured RNAs. So what we can then do with this when you compare it is we can kind of look at the change from all 3' UTRs to this helicase dependent, and we can see that it maximally separates around 0.3 in terms of overall structuredness. And what also is we don't really see any data below 0.2, so when it's below 0.2, wild type and the helicase mutant can both bind there. So what we can then do is what we did is we classified it as anything above 0.3 is highly structured, anything below 0.2 is poorly structured, and we just kind of leave that middle group alone since we don't really know what to call that. So then what we can do is we can ask, can UPF1 preferentially regulate the highly structured UTRs compared to the poorly structured UTRs? And to do that, I first chose 14 candidate UTRs, which had 7 highly structured and 7 poorly structured. And because UPF1 is thought to be length dependent, I made sure that all 14 of the UTRs had very similar lengths, around 600 nucleotides. So now from there, what I can do is I can generate cell lines. And to do this, I'm using this DLD1 colon cancer cell line where I can stably knock down UPF1 using a lentiviral infection, in which I use 2 different shRNAs targeting UPF1 at different points. And then I isolate 3 single-cell colonies from each one, and then I can do RT-qPCR and triple kits. So that way I have some statistical significance when I perform these assays. So what I found I was very excited to see is this heat map. What it's showing is the 7 highly structured on the left in the various columns, and the 7 poorly structured on the right in those columns. And then the 2 different rows are the 2 different shRNAs I utilized. And what you can hopefully appreciate is that when you look at which ones are being upregulated, it very selectively upregulated the highly structured UTRs while having no effect on the poorly structured UTRs. And then what I can also do is I can replicate this in a second cell line. In this case, this is a neuronal SHSY5Y cell line. And you can see that you see the very same results in which UPF1 can preferentially upregulate the highly structured 3' UTRs. And then the benefit of doing these stable knockdowns is I can now stably reintroduce UPF1 to confirm specificity and that this knockdown is due to loss of UPF1. So when I reintroduce wild-type UPF1 to these cells, you can see that the highly structured UTRs are reduced in expression, that this overexpression is lost upon reintroduction, where again, these poorly structured UTRs are unaffected by UPF1 status. What I can now also do with this is I can reintroduce UPF1 mutants. So the 2 mutants I reintroduced was this R615A mutant, which prevents UPF1 from binding to the RNA, and the DEAA one, that's that helicase mutant I've been talking about previously. So when I introduce these 2 mutants, you can see that there is no longer that reduction in the highly structured UTRs. So this is suggesting that in order for UPF1 to be regulating these, it needs the ability to bind to the RNA as well as unwind that RNA. So the next question we assumed was what other proteins are involved in this pathway? And so because, like I said earlier, UPF1 is involved in nonsense-mediated decay, we assumed this was part of the nonsense-mediated decay pathway. However, what we found was when we knocked down 4 other nonsense-mediated decay factors, you can see you're not seeing that nice pattern I was showing you before. You kind of see maybe some fluctuations, but they're not very consistent and reliable. So this is suggesting that the NMD factors are not in fact regulating these highly structured UTRs, and that this must be through some other UPF1 pathway. And on the right, I'm just showing 2 nonsense-mediated decay factors. So this is just showing that indeed I did properly knock down these factors. So UPF1 is actually found to be associated with a few other RNA decay pathways. But what we found was when we looked at those, both the stop-mediated decay with STAL1 and STAL2, as well as reginase-1 mediated decay, you can see again I didn't really see that phenotype in which when I knock down these other factors, we're not seeing any change in these highly structured UTRs, but we are seeing an upregulation of their known decay factors. So this then leads to the question is how is UPF1 doing that? And there must be some other proteins involved. And so to do that, I kind of moved forward, and luckily, this is a great tale of using publicly available data sets, but I was able to look at some mass spec studies in which they looked at what proteins are interacting with UPF1. And one protein really stood out to me, and that was G3BP1. And the reason it stood out to me was it has some poorly defined in vitro functions, such as it can bind to double-stranded RNA, as well as be an RNA helicase, which is very similar to UPF1. And the other reason I was very interested is it's been shown to be an endoribonuclease, so it's possible that this is the protein actually involved in degrading the RNA. So based on these findings, I wanted to move forward with this and see does G3BP1 also bind to these highly structured 3' UTRs. So again, using more publicly available data, is I was able to look at where G3BP1 is binding, as well as looking at the overlap, in which you can hopefully appreciate in this Venn diagram that there's a pretty nice overlap between the helicase-dependent UPF1 bound transcripts, as well as with the G3BP1. And so when you look at the cumulative distribution frequency here, you see a smaller shift with G3BP1 alone, but when you look at the transcripts in which they're both binding, you see this very nice strong shift to the right, which again is suggesting that when UPF1 and G3BP1 are bound, those tend to be bound to ones that are highly structured. So performing those similar assays I've shown earlier is I can knock down G3BP1 in these two cell lines, and you see this very nice upregulation again in which only these highly structured DTRs are being upregulated and the poorly structured DTRs are not changing at all. And again, reintroducing wild-type G3BP1, you see the same effect. The highly structured DTRs go down. And this time, the two mutants I chose is I deleted the two RNA binding domains, as well as I made the serine mutation, which you can see when you make these mutations, it no longer works. And the serine mutation is a very interesting mutation because the serine's under very hot debate right now, but has been thought to be involved in that endoribonuclease activity I talked about. But it's also possibly involved in stress gradient recruitment, nuclear localization, and a whole mess of things. So there's a lot of debate going on in terms of what this serine really does. But it has been, everyone is generally confirmed that it is heavily phosphorylated, so there's a chance it is doing something. And what we think that something is regulating the decay of these highly structured DTRs. Next is, since everything's been using these publicly available ECLIPSE-seq data sets, is I just wanted to make sure, are these two proteins really associating with the highly structured DTRs? So I was able to do some RNA immunoprecipitations, in which I pulled down both wild type and the RNA binding mutants of both UPF1 and G3BP1. And so when you look for enrichment for binding for wild type versus RNA binding mutants, what you see is that UPF1, like I've shown earlier, is wild type UPF1 can bind to everything. So you see UPF1 is mostly associated with everything. But G3BP1, we're very excited to see that it was only preferentially associating with those highly structured DTRs and not binding to those poorly structured DTRs. And then what we wanted to then do was see, can I get this separation, that if I mutate that helicase domain and repeat this RNA immunoprecipitation, will I see that association? And this is just showing that, again, we do see that association. That when you mutate that helicase domain, you no longer see as great of association, and that the wild type UPF1 can associate with the highly structured DTRs better than when it doesn't have the helicase activity. So because everything's been interacting with the same RNAs, one thing we had to ask was, are these working together, or are these just two separate pathways that happen to be regulating the same transcripts? So what I could do is I took the G3BP1 knockout cells and then did additional stable knockdowns. So the G3BP1 knockout cells were generated through CRISPR-Cas9 to get stable knockouts, and I did the lentiviral knockdowns with either the control UPF1 knockdown or G3BP2 knockout with CRISPR-Cas9. This G3BP2 is another homolog of G3BP1, which I'm not going to talk about anymore, but I'm just including it here. But what you can see is that when you don't have G3BP1 present, it doesn't matter if UPF1 is knocked down or not. And that without G3BP1, UPF1 is not having any effect. So this is suggesting that G3BP1 is essential, and that these proteins are most likely working together to regulate these highly structured UTRs. So just kind of concluding this first part, is so far what I found was that UPF1 and G3BP1 are regulating these mRNAs with highly structured UTRs, and this is independent of the previously characterized pathways. Additionally, what we found is that it's dependent on the RNA binding capabilities of both these proteins, and that helicase activity of UPF1 is very important for this mechanism to happen. So then moving on to RNA decay, everything I've been showing you so far has been steady state levels, so I need to first really show you that it's through decay and not through biogenesis. So to do that is I can treat my stably knocked down cells with the actinomycin D, which inhibits transcription, and then I can compare the half-lives for both either the G3BP1 knockout versus wild type and the UPF1 knockdown versus control. And then what ideally you'll see is that when you knock down these proteins, the highly structured DTRs, as you see here, increase. That's showing that when you don't have these proteins present, these RNAs aren't really degrading, and that this is through degradation. But then to look more, instead of just looking at these few examples, we thought it was about time to do some transcriptome-wide RNA sequencing to see, is this decay happening genome-wide, or is it just these 7 candidates? So just to orient you with what the RNA sequencing I'm showing you is about, is I have my DLD1 cells with G3PP1 wild type or with G3PP1 knocked out. And then I either treated it with actinomycin D for 18 hours or I didn't. And then from there, what I can do is I can get the steady state levels as well as changes in the decay rates. And what this is showing is this is just all the genes that are either upregulated in the knockouts or downregulated in the knockouts. And then what we found, what was interesting, is that absolutely all of these genes had longer half-lives when G3PP1 was knocked out, and that none of them had shorter half-lives. But now what we can do is, now that we have this large pool of genes that are both upregulated when G3PP1 is knocked out, as well as have longer half-lives, to see, are these genes with highly structured UTRs? One problem with RNA sequencing is you can't always properly identify which transcript they're coming from, just what gene. So for the next analysis, I'm only looking at genes that every single 3' UTR it has, it's always highly structured or always poorly structured. So when you look at that, you can see I'm losing some numbers when I do that, but what you can see is there's this very large enrichment of highly structured genes with only highly structured UTRs that are upregulated, as well as having the longer half-lives. And the ones with the poorly structured UTRs, you can see there's very few of them. So this is suggesting that indeed we're able to see that this pathway is working genome-wide, which is very exciting. But what is also very interesting is why are these poorly structured UTRs being regulated? So one thing to kind of confirm, first what we had to do is just confirm this. So just by doing some qPCR, we were able to show that indeed the genes that were upregulated in the knockouts are upregulated, as well as the genes that are downregulated are indeed downregulated. So then what we wanted to do is confirm, because we know they're being upregulated, is this through direct interactions or are these just indirect effects that we're observing due to this RNA sequencing? So I could go back to those RNA immunoprecipitation samples I showed earlier, and what you can see is the genes that are upregulated in the knockouts, both the highly structured and poorly structured UTRs, they're all being bound by G3P1, whereas the ones that are downregulated are not bound by G3P1. So whatever is causing this effect is not through G3P1 binding, but what's interesting is that, yes, the highly structured UTRs are binding as we would predict, but also these poorly structured UTRs are being bound by G3P1,